Today's topic is cellular respiration. Cellular respiration is a series of metabolic chemical reactions that extract the energy from the chemical bonds in the glucose and convert this energy into the ATP, energy storing molecule. In here shows steps of the aerobic respiration. It have three steps. Glycolysis, which happening is cytosol. Second, citric acid cycle happening in the matrix of mitochondria. Oxidative phosphorylation have two parts: A, electron transport chain, and B, chemioosmosis. Oxidative phosphorylation will happen close to the inner membrane of the mitochondria. How you can see in here, during the glycolysis, glucose converted to pyruvate. Then pyruvate will be passing through the outer membrane of the mitochondria in matrix, jelly-like substance, and this is where citric acid will be performed. Next, it will be moving whereas on the inner membrane of the mitochondria, whereas oxidative phosphorylation will happening, electron transport chain, and chemiosmosis. Electrons transfer energy in the cell. Energy conversion with a cell involve oxidation reduction reactions. They are together, occur simultaneously together. During oxidation reaction will remove an electron from the atom. And during reduction reaction, electrons will be added to another atom. In the cell, it's important to produce energy in the small amount. So it's not will be producing too much heat, and it's not will be that all molecules atom will move very quickly and damage the cell. So the transfer of energy in form of the electrons allows cell to transfer energy and use it in the small packages amount rather than in the one single large and destructive burst. During oxidation happen replacement of electron in form of the hydrogen ions on oxygen. During reduction happen replacement of the oxygen on electrons in form of the hydrogen ions. Electrons are removed from carbon as in methane in here, and it's oxidized during step of the reaction to carbon dioxide. During this oxidation reaction, hydrogen ions will be removed and replaced by the oxygen, how you can see in here on the image. The loss of electrons in form of the hydrogen ions is also happen by loss of the energy. The more hydrogen are removed, the more energy the specific molecule is lost. So methane will be having high energy because we have a lot of hydrogen ions, and carbon dioxide will have low energy because no hydrogen and we have only oxygen. And in opposite reaction, reduction, electrons again, if we convert carbon dioxide to the methane, what happening? We replacing oxygen on the hydrogen. And during this process, low energy carbon dioxide become high energy methane. And during this process, reduction, what happened? During reduction, molecules will gain electrons because they gaining hydrogen. And the more hydrogen, the more energy. So I'm adding energy in here. Electron carriers. Electron carriers have important function. They like electron shuttles, like a buses. They can accept electrons, electrons can attach to them, and then they can carry electrons from one molecule to the next molecule during metabolic reactions. And what's special about electron carriers that they can be easily reduced or accept electrons or oxidize loose electrons. For our course, you need to know only two examples of the electron carriers, NAD plus and FAD plus. How you can see they both vitamins B3, B2, and they're important for the energy. So more vitamins B you have from the food or supplements, the more energy you will have. So in here reaction, this equation show how NAD is working. So we have substrate, for example, glucose, and it's represented by this R. H indicates that glucose contain energy in form of the hydrogen. And if electron carriers NAD can be used in the reaction, so in this reaction, what happened? From glucose, hydrogen will be transferred and attached, bind to the NAD. And NAD will be reduced because it's accepting hydrogen, become NADH. And substrate become product, and the product will be oxidized. Oxidize means hydrogen replace on the oxygen. So in here, just not showing. Next, what happened? In the other chemical reaction, NADH, which electron carrier and carry currently electrons in form of the hydrogen, will be transporting these hydrogen ions together with electron to the other substrate. On the equation show R in here second. And what will be result of this? Substrate now will become product and it will be reduced. Reduced means it's gaining energy in form of the hydrogen. And our electron carrier NAD become in original reduced form. So it's, in oxidized form without hydrogen. During ATP-ADP cycle, energy will be uptaken or released. And ATP, it's like rechargeable battery, think about. It can uptake energy and release when it's needed. During hydrolysis, chemical reaction, this is when ATP will be break down to the ATP. And phosphate group, last phosphate group especially, this is bond between phosphate groups. This is where its energy will be located. Phosphate groups, they all have negative charge, so they repel from each other, not attracted. And it requires energy to connect them together, or it will release energy if we break down connection between phosphate group. And reactions when we converting ATP to ADP, from ATP to ADP called dephosphorylation because we're removing phosphate group. De mean a removal of phosphate group. So from ATP, whereas have 3 phosphate group, we will remove 1, dephosphorylation, and energy will be released during this process for different chemical activities. And when opposite happening, when we're converting ATP to ATP, we're using phosphorylation reaction because we're adding phosphate group to the 2 groups of in the ADP molecule, And because it's a synthesis reaction, we need to synthesize, produce new chemical bonds, so energy is required. ATP and energy. So almost all cells in the living organism on our planet have ATP, and most of them using glucose, fructose, or galactose metabolism. All these three molecules have the same chemical formula. They have 6 carbon, 12 hydrogen, and 6 oxygen. But all these glucose, fructose, galactose, they isomers. It means how exactly these all atoms attached is a little bit different. And this is why they call isomers. And ATP is connection between exergonic chemical reactions, mean reactions which release energy, exa mean outside, and endogonic chemical reaction which require energy, endo mean inside. So in the endogonic chemical reaction, it's reaction of the synthesis or moving something or opening channels. So all this require energy, it will be. An exergonic reaction releasing, so it's a type of the catabolic reaction will be. And ATP, this is what carry energy and giving or uptaking between these reactions. Please note that some bacteria use different substrate to produce energy. So it's not always glucose, fructose, galactose. It could be different substrate. Okay, so now we will talk about substrate phosphorylation. During substrate phosphorylation, substrate molecule, for example, A, will be phosphorylated. Phosphorylation means adding the phosphate group. And in the cells, it's use ATP molecule, for example, for phosphorylation. We can break down ATP molecule, mean we can break chemical bond between phosphate groups in the ATP. And phosphate group will be connected with new chemical bond to the product. And kinase, it's type of the enzyme which performing this phosphorylation. And during this reaction, from ATP, we will produce ADP, and phosphate group from the ATP will be attached to substrate. And because now it will have phosphate group, it will be completely different molecules. B. So this will be a product. And in between, what missing in here is a complex when enzyme connected to both substrate A and ATP phosphate group.